Hello friends, welcome to Cosmos Study Circle. Continued from the previous chapter. If you not previous video, then watch first all previous video, after then watch this video.
Today under cellular organization we will discuss about the cilia and flagella, microtubules, microfilaments and centriole. First have a look at the cilia and flagella. Okay. Many cells have long and thin, about 0.2 to 0.3 micrometer cytoplasmic structure extending from the surface of the cell are known as cilia or flagella. Cilia are short motile organelles generally present in large numbers covering the surface of the cells. Flagella are similar but larger and lesser numerous as well as more complex in their movement. Cilia and flagella fundamentally have the same origin and structure. Both cilia and flagella grow out of the basal body. Ultrastructurally, the cilia and flagella consist of 9 outer fibrils around the 2 larger central fibrils identical to microtubules in their construction. Each outer fibril consists of 2 microtubules. In both cilia and flagella, the entire assembly is seeded by a unit membrane which is simply a part of the plasma membrane. Cilia and flagella are usually locomotory organs. However, many animals have cells, the cilia of which serve simply to move materials past the cell. Flagella are relatively longer and cause cell movement. Cilia are relatively smaller and work like oars, causing cell movement or movement of surrounding fluid or substances inside the organs. Exonemes possesses 9 microtubule doubles along the periphery and a pair of centrally located microtubule. Such an arrangement of microtubule is called 9+2 array. The central tubules are connected by bridges and are also enclosed by a central sheath. The central sheath is connected to one of the tubules of each peripheral doublets by a radial spoke. So there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Cilium and flagellum emerges from centriole-like structures called the basal bodies. Alright, the second topic is microtubule. The microtubules are a straight tubular structure, usually about 250 angstrom in diameter, but as thin as 150 angstrom filaments have also been observed. They are extended for several microns and they are stiff and provide some rigidity to those parts of the cell in which they are located. Microtubules have dense wall, some measuring about 40 to 70 angstrom thick, surrounding a less dense central area. In addition, each microtubule appears to be surrounded by a slightly staining region about 100 angstrom thick, from which organelles and particulate matters of the cytoplasm are excluded. The ultrastructure of microtubules was first of all described by Potter and Leadbetter in 1964. Have a look at the function of the microtubules. There are mainly 3 functions of microtubules. Number 1, microtubules play important role in cell division. Number 2, microtubules are used in construction of flagella, cilia and centrioles. Number 3, microtubules transport the ions, water and small organic and inorganic molecules. That's all about the microtubules. Have a look at the microfilaments. Microfilaments are very short terms or cell organelles. Microfilaments are fine thread-like fibrils, some 40 to 60 angstrom in diameter. They are believed to be capable of contracting and forming masses of network at various places in the cell. But elsewhere, according to Peter Mann in 1964, microfilaments are made up of one of the major protein components, maybe actin or muscle cells. According to LeBlanc, microfilaments have been shown to be involved with movement of the cytoplasm called amoeboid movement or cyclosis or cell steaming, depending on what type of cells are observed in. That's all about the microfilaments. Have a look at the next topic, centriole. Lower plants, some microorganisms and animal cells contain two membranous and fibrous bodies near the nucleus in the cytoplasm. These are called centrioles. Each centriole measures about 0.2 micrometer in diameter. Centrioles were first reported by Bendon in 1887. Ultrastructurally, the centrioles are cylindrical elements having a structural plan essentially like that of basal granules and cilium. Centrioles are generally paired, the two cylindrical elements always being oriented at right angles to one another. Each centriole is composed of 9 peripheral bundles of microtubules instead of 2 as in case of cilium and the 2 central microtubules. The peripheral bundles are actually the extensions of the centriolar grouping of microtubules. Centrioles are center for spindle fiber formation during cell division. The spindle fibers are formed of microtubules of 230 to 270 angstrom in diameter. They also form the basis of cilia and flagella. Many visual receptor cells and retinal rod cells are originated from centrioles and cilia-like bodies. Centrioles in some way can initiate and control the formation of fibrous protein structure. That's all about centrioles.
साथियों, वीडियो की नोटिफिकेशन पाने के लिए चैनल को सब्सक्राइब कर ले। अगर आपको कोई और जानकारी चाहिए तो नीचे दिए गए नंबर पर कांटेक्ट कर सकते हैं। जल्द ही मिलते हैं फिर एक नई वीडियो के साथ।